Hello and a very warm welcome to everyone joining us for today's Select Science webinar entitled Cell Counting Method Development for CAR T-Cell Therapy. My name is Dr. Lois Manton-O'Byrne and I will be moderating today's presentation. I am delighted to introduce today's speaker, Dr. Leo Chan, who is a Technical R&D Manager at Nexcelom Biosciences. Just before I hand over to him, I would like to mention a couple of things. Please feel free to ask any questions for the Q&A session at any time during the webinar by clicking on the Ask button at the top right of the auditorium. You can view the presentation in full screen by clicking on the tab at the bottom right of the window player, and the on-demand link will be provided a few days after the webinar. At the end of the webinar, you will also be able to download a certificate of attendance and learning outcomes summary. These can be found in the Resources tab on the webinar player. Without further delay, I am delighted to hand over to Leo, and I thank him for presenting to us today. Please go ahead, Leo.
Hi, everyone. My name is Leo Chan from Nexcelom Bioscience, and thank you for coming to the Select Science webinar series with the title Cell Counting Methods Development Process for CAR T-cell manufacturing. And today I would like to go through how to develop cell counting methods and what are the pitfalls of different cell counting methods that's currently being used for CAR T-cell manufacturing. And I hope by the end of the webinar, you will understand the importance of cell counting in these type of field and development and manufacturing. So to begin with, cell counting, that is a very important topic, but who cares? And cell counting is actually a very trivial point in a biological experiment. It's oftentimes overlooked due to its minuscule portion in a biological assay. However, it is actually very important now in terms of all the cellular therapy progress that we have made in the field. So, as we know, FDA, NIST, and ISO, they're all working together to standardize cell counting methods for cell therapy. So they have put together different cell counting workshops to share practices in cell counting measurements, as well as putting together ISO standards for cell counting. Part 1 and Part 2 have already been released, and they're also currently working on multiple parts that would be improving cell counting methods for this particular field. So from us, Nexcelom Bioscience, we have worked with multiple clinical institutions to improve cell counting and viability accuracy beyond the use of Trypan blue. So there have been multiple publications published on this type of method using automated cell counters. So such as MD Anderson looking at T-cell using the Sleeping Beauty expression to look at cell counting and viability, as well as TCR transduced T-cells, and finally looking at T-cell engineering. All of these cell-based assays require a very accurate and rapid method to look at cell count and viability. So we know there are multiple steps during CAR T-cell development and manufacturing. So during each step, cell counting is very important. So if we look at the first step where the apheresis occurs, so we would need to identify a good patient sample, and a good patient sample is collected. Second, we will need to know the amount of viable T cells after gene editing. Third, we will need to also verify T cell proliferation over time to make sure that T cells have expanded, and as well as understanding the T cell function. Next is to ensure cell counting or cell conditions and concentrations are proper for dosage. And finally, ensuring the gene-edited T cells are viable prior to infusing back to the patient. So in order to do cell counting and viability, the most traditional method currently is using Trypan blue and manually counting on a hemocytometer or automated cell counting methods. And as we know from a hemocytometer method, it is very time consuming and has operator dependent variation. Other side effects from using hemocytometer may also be blurry vision, as well as some headache that comes with it. So these are some of the issues that people often face when they're trying to do cell counting manually. So by using automated method, these can be removed. However, there are some issues using Trypan blue. First, if the primary samples have RBC contaminations, in this case using cellular therapy, there are definitely primary samples that have a lot of RBC contamination. So in an example bright field image shown over here, it is actually very difficult to count in the bright field. So you can see that there are multiple debris objects that's in the image, and there are some cells that look like RBCs or PBMCs. So because of these debris and RBC contamination, you can overestimate cell viability. So if you look at these particles with a red arrow, are they RBCs or are they nucleated cells? And the cells with a green arrow, are they PBMCs or are they debris? It is very hard to determine. The second issue is we have found over the years of working with many different users, Trypan blue effects on near dead cells or dead cells. So showing an image of a Trypan blue stain sample, you can see sometimes there's uncertainty in identifying the dead cells. So you can see that the green arrow is pointing to the live cell, the red arrow points to dead cells, but there's also this large diffuse object that we don't know what they are, which I will show later on in this presentation. So first, let's get back to the RBC contamination. So we know residual RBCs can cause overestimation of cell count using Trypan blue. Because we're using bright field, we can see that there's these cells with a red arrow, that's RBC. Also, these ones, that's the PBMCs. And on the graphs showing on the right, you can see that there are multiple different samples. Some have more and some have less of these RBCs that's in the sample. So in this experiment, we found that residual RBC is highly donor dependent. We manually counted 15 glycol processed PBMC samples that were all processed the same way. And we observed varying RBC contaminations from low, medium, to high. And these are all donor dependent, causing inaccurate cell count, which will also cause downstream assays to be inaccurately performed due to these numbers. So looking at how Trypan blue affects the dead cells, what we did is we took a flask of Jurkat cells and we left the Jurkat cells out on the counter over multiple days. And every day we would take out a sample to measure with Trypan blue and take an image. So at 0 hour, when the cells are first removed, we can see that the dead cells looks very dark and easy to identify. At 6 hours, the morphology starts to change. At 12 hours, you can see that the color of the dead cells stained with Trypan blue is getting lighter. And at 24 hours, it's even lighter. And over 48 hours, 72 hours, 96, 168 hours, you can see that because of these unhealthy cells, when they're stained with trypan blue, the membrane is effectively affected by how the trypan blue interacted with the cells. So these light blue diffuse objects are often not counted using hemacytometer in a microscope because you can't see them. So if we put a side-by-side comparison, in a freshly cultured Jurkat cells, you can see very clearly what's live and what's dead. When unhealthy Jurkat cells, for example, cells left outside for 168 hours, you can see live cells, but there's these diffuse objects. Do we really need to count these objects? And most oftentimes, they're not counted. So what we have done is, over the 168 hours, we measured and counted the cells manually using Trypan blue. We also use an automated method using the Cell-Meter Auto T4, using Trypan blue as well. And we also measured the sample using propylium iodide to identify the dead cells, as well as using something called acridine orange and propylium iodide to measure the live and the dead cells. So we used this method and counted the total number of cells over time within the 168 hours. So the total cell count, as you can see, if you're using a fluorescence method, using propylium iodide or acridine orange and propylium iodide, you can see the total cell concentration remain pretty much consistent over time. However, if you use Trypan blue, the number of cells decrease over time, which doesn't make sense. Then we further examine the live cell count. Over time, both live count using Trypan blue and fluorescence dye decreased in that 168 hours, which makes sense. The number of live cells should drop. However, if you look at the dead cell count, the number of dead cells counted using fluorescence increased more than double of the Trypan blue method, which is showing on the bottom of the graph. And this causes the cell viability to not decrease as fast as using fluorescence method. And we attribute that to the number of diffuse objects that's not being counted using Trypan blue, which causes the morphology to change and the colors to change, so oftentimes they're not picked up. And you can also see there is a plateau from 80% until about 75 hours using Trypan blue when the viability starts to drop. And you can see that because of this plateau, Trypan blue overestimates cell viability due to the less counted dead cells over time. So most of the time when you're trying to qualify cell counting and cell viability method, researchers would use heat-killed sample to test out the method. However, heat killing does not represent the morphology of naturally dying cells. If you heat kill, you suspend them in time where the membrane of the cells are still okay. And by staining with Trypan blue, it retains Trypan blue dye very well and gives a very dark color, which can be counted by manually counting or automated method very easily. However, if you have naturally dying cells, that's where the Trypan blue have the most effect. And most of the time when you have primary samples that have multiple effects, because there are a lot of different cell types that have different effects with the Trypan blue. So you can see live and dead cells that's easily counted. So by using the fluorescent nuclear live dead viability stain mentioned earlier, this can address these two issues raised from using Trypan blue. So you can see in the bright field, there are these RBC contaminations you can see with the biconcave morphology, and then also the PBMCs with the blue arrow. However, in the fluorescence, because using the actinic orange and propylene iodide, the live cells are green fluorescent positive, and the dead cells does not have the fluorescence in the green color. Only have the red color for the dead cells, whereas the RBCs, because they don't have nucleus, so they won't have fluorescence. So if we look at what AOPI is made of, AO is a permeable dye that can penetrate both live and dead cells. The AO will bind to the DNA and fluoresce green. The PI will only enter the dead cells, binds the DNA of the dead cells, absorbs the green fluorescence of the AO, which produces a bright orange and red color. And no signal is generated from non-nucleated cells and debris such as RBCs or platelets. This makes the cell counting much easier and works on primary samples where a lot of RBC contaminations, debris, and platelets are presented in the sample. So if we use this and look at apheresis or leukapheresis sample, the fluorescence method can actually count the live dead cells in these patient samples. By looking at this bright field image, you will probably count total number of cells with this brightly centered cells object. But if you put it in fluorescence, only 39 of them have a nucleus. So basically overcounting 80 cells, and that's not nucleated. So this is probably a 3 times overcounting of the cells in the sample. So another experiment that we ran is running 6 different leukapheresis samples, and we understand that overcounting ratio is actually sample dependent. Again, it's not a systematic trend. So we did manual counting using hemacytometer and Trypan blue. We did automated cell counting using cell-meter and AOPI. And then we used the 6 different leukapheresis samples, and we can see that out of the 6 samples, different levels of overcounting is achieved using Trypan blue and hemacytometer. By applying the fluorescence method to the 15 patient samples that we worked with in the previous slide, we can show that by using AOPI, we can actually count the number of nucleated cells comparable to manual counting, so not including the red portion where the RBC contamination is counted. And to validate AOPI against Trypan blue, the only way we can do this is by doing a total nucleated cell count. So what we had to do is actually lyse the sample and stain with methylene blue or crystal violet. Then we can do a total nucleated cell count using hemacytometer and microscope, at the same time comparing to AO and PI positive cells, which show that they're very comparable. That validated the AOPI method. So what we would like to do is apply fluorescence-based cell counting method, as well as bright field-based using Trypan blue for the CAR T cell development and manufacturing. And this is based off a principle of fit for purpose, because at different steps of the CAR T cell development and manufacturing, it may require different type of cell counting method. For example, if you look at apheresis sample where a lot of RBC contamination from the primary cell are presented, platelets and debris, you definitely require nuclear fluorescent stain such as the AOPI to count your cells to get an accurate cell count. Next, when you're doing transduction and transfection of the T cells or of the primary sample, you can use the fluorescent cell counter and count GFP positive and PI negative cells. And when you look at the expansion, since by this time, most of the T cells are clean and these RBCs and platelets have been removed, you could just use Trypan blue and AOPI to count the cells. And looking at functionality testing, you would need to have a baseline of the cell count. So again, using Trypan blue and AOPI would be possible. Same thing as for conditioning and dosing, using Trypan blue and AOPI is good. And finally, infusing back to patient into the patient, you can still use Trypan blue and AOPI since most of the cells are very clean at that point. So what we did is we went through both FDA and NIST recommendations on how to qualify cell counting method. And we have come up with utilizing their guidance, looking at different qualification parameters. So the first one is called robustness and ruggedness. This is where your cell sample, when you introduce non-specific particles. So for example, if you have PBMCs or if you have Jurkat cells, if you introduce beads or red blood cells, that should not affect your cell counting results. So that's one way to look at robustness and ruggedness. Second is looking at linearity. And this changes between different cell counting methods. Some cell counting methods have large dynamic range, some does not. But the point is you need to test the linearity of your cell counting method from very high to very low and identify the range that would best be suited for your cell counting method, as well as viability from 0 to 100%. Finally, you can look at accuracy with these type of cell counting method, which comes from the standard deviation when looking at the linearity method for identifying what is the range that would be best for the cell counting method. And again, range also falls into the linearity experiment. The intermediate precision is looking at different lots, different operators, or different days of using the cell counting method and look at how precise they are over these type of parameters. And just for precision is just repeatability, measuring the same sample multiple times and looking at the standard deviation. And at the end, we have LOD and LOQ that looks at the lowest cell density detected in a serial dilution or lowest cell density that's accurate and precise. And all of this falls under the linearity experiment that you can use to identify this lowest cell density. And again, we follow the FDA and this guidance for coming up with these parameters, and we have used this type of experimental settings and experimental procedure to test all of the cell-meter instruments that we have. So, what is the most important step when validating or qualifying your cell counting method? It is sample preparation. One of the major things that we have discovered over the years working with users is that sample preparation can cause 50% of the error when you're trying to do qualification, such as uniformly pipetting your sample. Make sure you vortex, make sure you follow the exact same procedure, not letting the cells settle. These are all very important points that we have discovered over the years to make sure that the cell counting procedure is properly done when validating the cell counting methods. In addition to cell counting, cell-based assays are also very important for CAR T cell development and manufacturing. So when we're looking at all these different steps, At the same time, cell counting methods can be applied. So for example, looking at these 4 different steps, you can look at immunophenotyping when you first collect the patient sample. You can look at transduction and transfection efficiency, T-cell activation and proliferation assay, as well as CAR T-cell mediated cytotoxicity or T-cell migration. So here I want to show an example of utilizing a plate-based image cytometer called the Cell-LIGO that can perform these type of functional testing on the cells directly in a plate. So an example for T-cell activation and proliferation measurement, you can use the Cell-LIGO to directly capture images of T-cells in a plate and stimulating them with CD3 and CD28. You can see the change in size of the T-cell, and using the Cell-LIGO software, you can count the activated T-cells over time. And by counting the activated T-cells, you can monitor sample that's unstimulated versus sample that's stimulated that shows where the T-cells can grow to over 140,000 cells in a well. Second example is looking at CAR T-cell mediated cytotoxicity. This is where you can label the target cells with fluorescent protein or fluorescent dyes that can be monitored over time. So for example, this is an experiment where the blue fluorescent protein expressing cells, the target cells, does not have the target antigen that's on the surface, where the pink color does have the positive antigen. And in this experiment, you can see samples with the irrelevant CAR T, the target cells remain in the well over time for about 24 hours. In the target CAR T, you can see the target pink colored target cells are killed off where they don't exist or are not presented in the samples anymore. And by counting the number of cells, you can show that with a normalized cell number, at time 0, all of the target cells remain. But at 24 hours, you can see the 4 different CAR Ts show significant reduction in the number of cells compared to untransduced CAR and irrelevant CAR, which indicate cell killing over time. And this is done directly using the Cell-LIGO image cytometer to directly count the cells in the well. So at this point, we are continuing to work with NIST to develop standardization for cell counting. They have already published a paper on how to qualify cell counting measurement. So we're now working with them on looking at utilizing their procedure to qualify our cell counting methods, as well as developing cell viability qualification methods, which are significantly more difficult than just cell counting because of debris, because of changes to cell morphology by Trypan blue, as what we discussed earlier in this presentation. So the takeaways for what we have done in the past 13 years is that cell counting is an important step for CAR T-cell therapy that is often overlooked. Trypan blue is the traditional method for cell counting, but has 2 inherent issues. First is RBC contamination, which can overestimate cell count and viability. Second, Trypan blue affects dead cell morphology, which can underestimate dead cells and overestimate viability. Next, using nuclear fluorescent staining such as the AOPI on cellometer can eliminate these issues caused by Trypan blue. Next, there are guidelines to qualify cell counting methods, which we have adopted for our technologies here at Nexon Bioscience. And cell-based assays can be performed in a high-throughput plate-based format, improving efficiency for CAR T-cell development and manufacturing. Thank you for your attention for today. I hope you have any questions. You can please ask them, and I'll be happy to answer them today. Thank you.
Thank you very much, Leo, for an interesting presentation. Now let's move on to the last part of today's webinar, the Q&A session. So we've had lots of great questions coming through, so thank you very much for that. The first one, does AO bind to nucleotides that could be present in the media after the cells lyse/long cultures? And if so, how do you correct for that background?
That's a very good question. So it says, does AO bind to the nucleotides? Well, we have seen it before, but usually it's right after cell lysing, you can still see free nuclei flowing around. But if you're doing a long culture, what happened is we have also seen that the AO signal often decrease, so that signal becomes very dim. So there's a difference between that signal and the actual cell. And what you can do is you can also gate those small objects out by size gating using the ceilometer, so you don't pick those objects up when you're trying to count the cells. So that's the answer. If there's something that's unclear, please reach out to me after the webinar.
Fantastic. Thank you very much. The next question, how do we properly prepare cell samples to perform the qualification assays? So one of the slide in the presentation was about sample prep, right? Because there's a lot of different parameters for doing the qualification methods. It is important that you have proper sampling. So, for example, if you're testing just a regular cell line, make sure you have enough cells so you can go, when you're testing your qualification method or your cell counting method, you can go from a very high concentration to a very low concentration, so you know that you have good dynamic range on your cell counting method. In addition to that, in the presentation, I mentioned that sample prep is very important. What I mean is, when you're getting the sample out from the flask, from the tubes, you have to make sure that every single step is consistent. So if you're pipetting up and down 10 times, keep it the same. And make sure that every time you pipette, the sample is mixed properly, it's vortex. So you're always getting as uniform of a sample as possible when you're doing the next pipetting step. Because from my research in the past, it seems that there's multiple publications that says pipetting is the #1 error from doing these type of qualification. If your sample preparation is not done properly, then you're going to have inconsistency, variation when you're doing these tests that may disqualify your qualification or your counting method. However, depending on how your operator is doing the testing, you have to look at all the parameters, all the results that come out, and then look at what is the best, what is an acceptable criteria for our facility, for our institution, for our CAR-T process. What we have been doing is we have been generating a document for these qualification methods, looking at all these different parameters. So we're hoping to have a document ready on our website that you can download probably in the next couple of weeks or so. Great, thanks, Leo. Lots of great questions coming through. On here, we love our K2, that's great. In our CAR-T process, we find that cells that are cultured for 6 to 8 days don't take up AO very well. Have you observed that as well? And any guess as to the physiology that drives that process? Thank you.
That's a very good observation. We have seen that in the past. So there's a couple of things that may be happening. One is after a long culture time, sometimes the pH may shift a little bit. That may affect the uptake of the AO. So that's one thing. We are working on another dye similar to AO that may take up that will take up much more nicely than AO and stain much more brightly and more localized. So that stain, we're still doing a lot of field testing to make sure that it works on all sorts of cell type. And once that's ready, we will communicate this to our end users for that dye. It's called nuclear green dye. It's actually on our website right now that you can use. And we have a couple of colleagues that have already tested with end users that's doing T-cell culture. So that's one possibility. The other possibility is what we have seen with other users is with these long cultures, there are these objects that actually look like cells that's formed. They may not have a nucleus. So what we did is we did AOPI staining, and we did Hurst and PI staining. So all these are nuclear dyes. And we got similar cell counts with all these nuclear dyes. Then we did a manual counting of the bright field and Trypan blue. And what we got is we got more cells, more objects counted. Those objects that are not taken up with the AO or the Hurst dye, they don't have any, it seems like they don't have any nucleus. Then we did another stain called calcium AM to look at enzymatic staining. Those objects that don't have nucleus are enzymatically active. So we were labeling those objects ghost cells because it seems like they don't have a nucleus, but they are enzymatically active. So at this point, some of our end users are shifting towards only counting the real nucleated cells and disregarding the ones that don't have it. But some of our end users are still including those cell-looking objects that don't have nucleus to stay consistent with what they have been counting.
Fantastic, thank you. Another question. Are there other problems with Trypan blue staining?
So with the two issues that I raised, what we found in the field was one is the RBC contamination in the bright field, and the second is the morphology change from the Trypan blue. What we have seen is other issues with Trypan blue is, of course, the storage has a problem, right? So if you leave Trypan blue for a long time, it starts to form crystals that's in the solution. So oftentimes you have to filter, right? Otherwise, when you stain with the cells, you do see those crystals that may look like cells that you mistakenly count those. So there's some, more logistic or maintenance with the Trypan blue dye. Some other observation we have seen is besides the overestimating the live cells or underestimating the dead cells, is that oftentimes when you're doing hemocytometer counting, if you have a lot of cells, then it takes a long time to count them. Then over that period of time, the Trypan blue will have a lot of effect. Maybe you can't count it within 5 minutes, but even with that 5 minutes, it does affect the cells. And if you're counting something like very sensitive, like hepatocytes, and if you have we have seen within the first 2 minutes, trypan blue can kill hepatocytes and causing the viability to drop by 10 to 15%. So those are some of the issues with using these type of staining method.
Thank you. Really interesting. The next question we have, lots of great questions coming through still. Is AOPI stain toxic to the cells?
So AOPI, currently what we have observed is it is a nuclear binding dye. So of course you cannot leave it with the cells for a long time. However, for our testing, we have done staining of the cells for with AOPI for up to 2 hours, and the viability dropped from, I think it was about 85% to about 80%. That's within 2 hours. So what we usually recommend is if you have a lot of samples, so let's say if you need to count maybe 20 samples. So what we recommend is just stain for 5 samples first, put them into the counting chamber and count them, and then do the next 5, and then do the next 5. So to eliminate that type of uncertainty, because there are differences between different cell types. So if you're doing primary cells, they may be a little bit more sensitive than doing staining cell lines. But what we have seen is within 2 hours, there's not too much change in viability.
Thank you. Thanks for that, Leo. Unfortunately, that's all we've got time for today. Thank you very much for today's informative discussion and presentation, and thank you to everybody for joining us online and for your interesting questions. There were loads of them, so that's great. I hope that you found this a worthwhile session. If we didn't manage to answer your question, we will follow up with you personally. If you have any more questions, please feel free to email me at editor@selectscience.net. So that's editor@selectscience.net. And if you would like to listen again to today's webinar or invite a friend to listen, it will be available on our website in the next few days. Goodbye, and thank you once again for joining us. Bye.